Okay, so our next speaker is Dr. Stephen Collins for the Farncombe Family Digestive Health Research Institute and McMaster University. And he will be telling us today about microbiome interactions with the gut and the brain.
So, good morning, and first my thanks to the organizers for giving me this opportunity to participate in this. That's what I'm going to talk about, the sponsorship. This is my bio. I was a medical student once, so I take some of these comments to heart, and I still practice gastroenterology as well as doing research. I have no disclosures that are relevant to this talk. So in the next few minutes, I want to cover a fair amount of ground. I want to give you an overview of the microbiome, talk about how this microbiome, how this system influences intestinal physiology. Primarily in health initially. Then to talk about the interface between this organ system, the microbiota, and the gut-brain axis. And then at the end to talk about how the microbiome might impact on our understanding and possibly treatment of disease. So this is a rapidly moving field. Current knowledge and the expectations for the future really do rely on active research that's currently going on, and a lot of it's in animal models. And it remains to be seen, whether all of this will transpire into a change in how we manage patients. So this community within us, the microbiome as we call it, what is it? Well, it contains primarily, but not exclusively, bacteria. And loosely speaking, when we talk about the microbiome, we're talking about the bacterial microbiome. But there is the virome, which is the virus population in the gut, the majority of which are phage that reside often within bacteria, modifying their behavior, their function by integrating their DNA, their genome into that of the bacteria. And that's the majority of these viruses. The relationship between bacteria and viruses in a quantitative sense is about 1-to-1 ratio. And now this is the microbiome, which are the fungi that are present, mostly Candida and Saccharomyces. They comprise about less than 0.01% of the total microbiome, but there is increasing interest in how these fungi can influence the bacteria and how they can influence host. And there's been some preliminary work regarding this in the context of inflammatory bowel disease. So this is an area to watch. And in addition, depending on where one lives and what one does, parasites and protozoa also constitute part of this collective microbiome. So not just bacteria is the message. Some terminology to get straight at the beginning. The term microbiome really refers to the collective genome of the community. And as I said, loosely speaking, it refers to the bacterial, but it can, of course, refer to the other virome and microbiome in the gut, too. Microbiota usually refers to bacterial organisms themselves, their function, their metabolic activity, and their interaction with the host. This term dysbiosis is a very loosely used term, and essentially it reflects any kind of perturbance of the community, and particularly when it has an impact on host function. But a very loose term, and people use it for their own designs. Axenic, when we talk about mouse studies, axenic mice are mice that have no bacteria. And GNOTOBIOTIC mice are mice that are colonized with a prescribed bacterium, either a single bacterium or a restricted flora, but it's important to know the difference between these terms when you come to read the literature. For the last decade or more, we've seen an explosion of work profiling the microbiota, particularly the bacterial microbiome. And this is, more often than not, has been done using 16S ribosomal RNA sequencing. This sequencing depends on this hypervariable region that can provide, specific identification of bacteria and also of archaea, which are very primitive pre-bacterial organisms that survive very, very extreme conditions on this planet. Whole genome or shotgun sequencing is an unrestricted sequencing approach that uses random primers, and as a result it detects pretty much everything that's there, including, in addition to bacteria, viruses, parasites, and fungi. And as we learn more about the virome and the microbiome, I think that understanding the changes in each of these domains is going to become increasingly important in understanding disease. And I think that this will overtake the use of 16S in the future as our knowledge improves. And the cost of this is coming down. Because in the past we could sequence more bacteria than we could culture, it was generally assumed that the bacterial composition of the gut is largely unculturable, and this turns out not to be the case. A couple of recent publications have demonstrated this using using a variety of culture conditions, primarily anaerobic, and then sequencing this using 16S compared to the sequencing of microbes or of samples coming just directly without culture and sequenced, that about 95% of the intestinal microbiota can be cultured. And the ones that appear in sequencing that so far have not been cultured really constitutes a very, very small proportion. So this is important then, because if you can culture these bacteria, it opens up a whole range of research that previously really wasn't thought possible. You can take these bacteria now, maintain them, and colonize mice or other organisms and conduct experiments that lead more towards causality than association. So I'm going to use this term microbiota from here on out to describe the bacterial component of the microbiome. So there were two huge government-sponsored projects in Europe, the META-HIT, and sponsored by NIH, the Human Microbiome Project, aimed at sequencing every single bacterium in humans. And this so far has identified over 2,000 species. They can be classified into 12 different phyla, and the dominant phyla, accommodating about 93.5% of these, are proteobacteria, firmicutes, actinobacteria, and bacteroides. And the majority of these are strict anaerobes or facultative anaerobes. So I'm sure you've seen these sorts of comments that the human intestinal microbiota contains trillions of organisms in the region of 10 to the power of 13 or 14 microorganisms. It was originally thought that these bacterial cells outnumber the cells of the human body by a factor of 10 or 100 to 1, but more recent data suggests that that's not the case, and it's more likely to be closer to 1 to 1 than the higher numbers. As far as the genomes are concerned, the microbiome, the bacterial microbiome, has probably 100-fold more genes than the human microbiome. Now, the majority of these organisms reside in the distal bowel, where they, as I'll tell you later, accomplish a number of objectives. But part of this is to process materials that are indigestible to us as humans, primarily but not exclusively for the benefit of the host. And at birth and throughout life, and this is a very important function, and I'll come to this in a minute, the microbiota informs the mucosal immune system. And as I'll show you in the rest of this talk, the intestinal microbiota exerts an influence on the host that extends well beyond the gastrointestinal tract to distant organs, and in this particular talk, I'll talk a little bit about the brain. So for the next few slides, I'm going to talk about the colonization process, because I think this is going to be, in the next little while, a very important topic in terms of the research that's going on that suggests that the origins of some diseases may actually occur very early in life, and reflective of a disrupted colonization process. So we all thought, or it used to be thought, that we were born sterile, and that, based on emerging data, seems to be not necessarily the case, at least in all people. That there is evidence that the fetus can become exposed to a maternal microbiota through a transplacental passage into the amniotic fluid, and that these bacteria can make their way into the intestinal tract of the fetus. The significance of this at this point in time is not known, but I'll show you in a little bit how experiments done during pregnancy have effects or consequences for the offspring, suggesting that this intrauterine component might be clinically important. At full term of vaginal delivery, of course, the fetus, passing down the birth canal, becomes exposed to maternal microbiota, and then the colonization, and the infant gut at birth is not so deprived of oxygen as it is later on. So the initial colonizers may not necessarily be strict anaerobes, but they can be facultative anaerobes, such as Enterococci and Escherichia. Then, as time proceeds, with the introduction of feeds, and with the intestine changing and becoming more anaerobic, you then get the firmicutes coming in, Clostridia, Lactobacilli, and Bifidobacteria. And as the child then starts to sample more of the environment, over the course of about 1 to 3 years, there is a progressive increase in the diversity of the microbiota to about 1,000 species, more resembling that of an adult. But the critical period is clearly early in life, and as I've mentioned, intrauterine exposure might be something that we need to consider in the future. So, in essence, then, over the course of time, shortly after birth, one sees an increase in the alpha diversity, or in the number of species present in the richness, and a reduction in beta diversity, which means less turmoil, less turnover, and that means more stability. So the overall process then is one of increasing diversity, increasing stability, and as you'll see, this results in immunological maturity. So the priority for these colonizing these pioneer microbes is really to induce the maturation of the immune system and the barrier system to protect the host. So these early colonizers are responsible for stimulating the production of mucus and the enhancement of the physical component of barrier function, for the formation of gut-associated lymphoid tissue, which is, as you know, a self-contained immune system, to encourage the production and secretion of secretory IgA by plasma and other cells. And with some of these bacteria, particularly, for example, bifidobacteria and short-chain fatty acids such as butyrate, to generate a counter or anti-inflammatory environment that further protects the reactiveness of the young host to any kind of luminal insult. And this early and appropriate and proper colonization process is really critical not only for the maturation of the mucosal immune system, but also for the physiological systems. Many of these are dependent initially on the maturation of the immune system. So what can go wrong early in life? And studies, epidemiological studies, have shown that there is a relationship between maternal health in terms of diet or exposure to antibiotics and the development in the offspring at some point of allergic disease, diabetes, obesity, and it's well known that the children born by cesarean section have a greater risk of developing atopic disease, such as allergy, eczema, asthma, and also obesity. So again, reinforcing this importance of a controlled colonization early in life with the appropriate microbes. Now, I'll come to the brain later on in the talk, but it's also been shown that the colonization process affects the integrity of the blood-brain barrier, which consists of vascular endothelial cells and tight junctions, and supported by endothelium and brain astrocytes on the brain side of the barrier. And that germ-free mice have increased permeability in the blood-brain barrier, and this is reflected by reduced expression of tight junction proteins that were described by the previous speaker. And this can be reversed by colonization of the offspring with a specific pathogen-free microbiota, a normal microbiota. So there's a vulnerability then in utero, if this data is correct, that in utero the brain is exposed. And this may be the reason why perhaps insults that occur early or during pregnancy, as far as maternal health is concerned, could influence brain development. And there are some data, and I'll just briefly touch upon these. This is a study that was published last year, where they took mice, pregnant mice, and exposed them to low-dose penicillin for 3 weeks, and studied the offspring. And 6 weeks following the birth of the offspring, they found that there was dysbiosis, that the composition of the microbiota was altered following this intrauterine and postnatal, 3 weeks postnatal administration of very low doses of penicillin. That there was alteration in the blood-brain barrier, that there was increased cytokine expression in regions of the brain, such as the frontal cortex, that there was altered social behavior in these animals. They seem to be more aggressive, less socially adept, and that this could be attenuated by the administration after birth of a specific lactobacillus, Lactobacillus rhamnosus. So again, suggesting that there might be an intrauterine vulnerability that could lead could have consequences as far as brain and behavior are concerned later in life. And then there's been the work of Gloria Choi and colleagues on autism-like behavior in mice. And a series of very elegant studies performed by this group were really based on a couple of very large epidemiological studies in Scandinavia that suggested an association between viral infection during pregnancy and the development of autistic-like behavior in children, simply an epidemiological association. So bearing that in mind, the studies that they did showed that if you could induce severe immune activation in the mother during pregnancy, you could alter the behavior of offspring. And it was found that this was mediated by the induction of Th17 inducing and IL-17 producing cells, and that this was critically dependent on the presence of a microbiota. You couldn't do this in the absence of a microbiota, suggesting then that this maternal immune activation was driven by changes in the microbiota, and the interest there was on these segmented filamentous bacteria. So this is an evolving field. There's a lot of interest in autism at this point in time and its possible relationship to the microbiota, and it's really studies like this that I think will drive more informed clinical studies in the future. So moving on then beyond the early days of life, what factors influence the microbiota throughout life? And they come from three sources. First of all, the hosts, changes in mucin production, changes in the secretion of antimicrobial peptides and secretory IgA, the amount of bile acid and the kinds of bile acid that the microbiota are exposed to are important. As I'll show you later on in the talk, stress, psychological stress, seems to have an impact on the composition of the microbiota. And then any conditions that alter the physiology of the GI tract, such as functional GI disorders, irritable bowel syndrome, that change the physicochemical properties of certain niches, are going to select for a different type of microbiota. And then, of course, any conditions that involve inflammation or activation of the mucosal immune system are going to change the microbial composition of the gut through a variety of these other mechanisms. Perhaps the greatest influence is going to be from the environment, and obviously exposure to antimicrobial drugs, dietary change, infection, surgery as it relates to the GI tract and also to the biliary system, and any drugs that alter intestinal physiology, such as proton pump inhibitors. It's been shown that that changes the composition of the microbiota, and antidiarrheal drugs that slow down transit, alter secretion, will select for a slightly different microbiota. And then, of course, there are changes in the intestinal virome, and changes in the intestinal microbiome will also influence the bacterial composition of the gut. A recent study just recently published from Israel, where they took over 1,000 healthy individuals from different ethnic backgrounds, healthy individuals, and profiled their microbiome, and the findings are really quite interesting, that host genetic differences were easily detected across individuals of different ethnic groups or ancestries, as you would expect, that the microbiome, the intestinal microbiome of different ancestries were in fact quite similar, which was, I think, a little unexpected, that significant similarities were found in the composition of the microbiota of individuals who were genetically or ethnically unrelated, but who shared a household, so that environment was really a dominant factor dominating the host genome. And further investigating those environmental factors, that over 20% of this variability was associated with factors related to drugs, diet, and body mass and waist-hip ratios, in other words, metabolic syndrome, obesity, and so forth. So therefore, the take-home message from this is that environmental factors seem to dominate over host factors in determining one's microbiota. And this is also supported by more recent animal work. So what about the influence of the microbiota on intestinal physiology? If you take a germ-free mouse and make an incision, excuse me, and open up the intestine, you'll see a big difference. In the germ-free mouse, the bowel is dilated, it's thin, the cecum is massive. It's the first thing that pops out at you compared to a very normal-looking appearance of the bowel in normally colonized mice. So how do we go about investigating the relationship between the microbiota and host physiological systems? So there are basically three strategies. The first is really to go to one extreme and to compare how function in a germ-free mouse, how that compares to the physiology seen in a normally colonized mouse. The second, that has probably more bearing to life, is really to take a healthy mouse with a normal microbiota, with a matured host system, immune system and physiological systems, and to perturb that microbiota with a number of different strategies and see what impact that has on various systems. And the third, which gets more towards causality, is to see whether you can adoptively transfer the phenotype in question simply by transposing the microbiota into a germ-free mouse. So the first strategy then is the comparison of germ-free mice with colonized mice. The way we colonize mice is either by gavage of microbiota or through cohabitation with conventionalized mice, or simply by putting them into a dirty cage where the bedding and everything else will sooner or later provide them with the microbiota of that environment. This is usually very reproducible. You usually see very clear-cut differences between a germ-free mouse and a colonized mouse, and it's usually an obligatory first step in determining whether a system is influenced by the microbiota. And you can use monocolonization or prescribed colonizations or polycolonizations. The disadvantage is that the success of this is critically dependent on the absence of contamination. So if you see a negative study, that is to say, no discernible difference between a germ-free mouse's profile and that of a colonized mouse, the first thing you'd worry about is whether that mouse was actually germ-free. And we've been through this several times. As I said, because a germ-free mouse is essentially an immature mouse, immature in most of its tissues, you can't really extrapolate this data to later on in life when you're dealing with a microbiota interacting with a mature host system. and that's why the second strategy becomes important. So it really is a model of microbiota-induced host maturation. It's a kind of a developmental model, seeing the role of the microbiota in this. Here's an example of, in this case, mucin production, producing goblet cells in germ-free mice, and then in mice colonized by a single Bacteroides species. And you can see that within 2 days, you begin to see an increase in the number of these alcium blue stained goblet cells, and this increases progressively as the colonization with a single strain continues over the next 30 days. The previous speaker elaborated elegantly on what constitutes the barrier system, and the barrier system really is influenced by the microbiota. There have been very few studies on the intestinal barrier system in germ-free mice. It's an area of controversy, but essentially you've got an immature immune system that isn't going to drive maturation of that barrier. Once you introduce the microbiota, then you get maturation of the immune system and this selective permeability, which is the norm of the barrier system. Then, of course, as Jennifer had mentioned, with infection or with dysbiosis for other reasons, you get activation of the immune system, you get impaired barrier function, and you get inflammation of variable degrees depending on the nature of the model. But it's been shown that with selected probiotic bacteria, there have been several studies of this, that you can correct an impaired barrier function with selected probiotic bacteria. So this is a potential therapy in the future for disorders associated with impaired barrier function. Just to emphasize what Jennifer had said in her talk, that when we talk about barrier function, it's really a multidimensional thing. We're used to talking about it in the sense of the integrity of the epithelial cells and tight junctions and so forth, but really, and mucin production, of course, but there is a functional component to this too, with the immune system and secretory IgA and the production of antimicrobial peptides. This really does constitute a much more integrated description of the intestinal barrier. And this is each of these components is influenced by the intestinal microbiota. 5-HT or serotonin is an important neurotransmitter. It has many other functions too in terms of behavior and physiology, transit, motility. Produced largely in the enteroendocrine cells of the intestine, including the colon, as well as in enteric nerves. And this study here shows that in germ-free mice, the amount of total of serotonin or 5-HT is reduced, and that it's brought up to normal levels if you colonize it with murine microbiota, shown on the right, or even with human microbiota. So the microbiota speak probably, in this case, I think, to the enteroendocrine cells to encourage the production of this important hormone and neurotransmitter. The integrity of the enteric nervous system is also dependent on the microbiota, and here we're looking at specific staining for excuse me, neural density here on the left, and for ganglia on the right, showing that the absence of the microbiota results in a reduction in the density of nerves and ganglia, and that, oops, oh, this one didn't go, that the spontaneous activity in terms of motility, which is dependent on the functional integrity of the enteric nervous system, is attenuated when the the microbiota are absent from the gut. So in the germ-free mouse, the enteric nervous system is really poorly formed, less dense than it should be, and this translates into altered motility patterns in the germ-free mouse that can be corrected by colonization. So the second strategy then is, I think, one that's more reflective of postnatal life, and it's the experimental induction of dysbiosis in a healthy, otherwise healthy mouse. And you can do this a variety of ways. You can do it by taking a cocktail of non-absorbable antibiotics together with an antifungal agent, because if you just give the antibiotic, you'll get a proliferation of the fungi in of the microbiome, which will have functional impact too. Or you can do it by dietary change, using either a beef-enriched diet or in a high-fat, low-fiber Western-type diet. So when you're doing when these studies are being done, it's very, very important to try to capture that dysbiosis, because once you've finished with the intervention, the microbiota will quickly revert back to what it was before. So you have a small window of opportunity, in our hands probably less than 2 weeks, depending on how you house the mice, before you can to see the change. So the advantages are you're dealing with a mature host, where the systems are mature and responsive. It's reflective of what we call dysbiosis in clinical or experimental biology. And you can do this with gene deletions. You can use knockout mice or knock-in mice if you want to study host effects, provided they're not to the extent that a significant change in the microbiota is going to kill the animal. So, for example, with mice that lack, for example, MYD88, an innate immune adapter protein, they can't withstand big changes in the microbiota. So there are some limitations. But the main disadvantage is that you can't easily transfer this model between labs, because the microbiota in different labs are different, because they're influenced by the environment. And so a given set of antibiotics will produce a different effect depending on what the baseline microbiota is. So if you're really looking at in well-characterized responses, those responses may be quite different between labs, simply because you're starting with and ending up with a very different microbiota. But it makes the point that if you change the microbiota, you change the system. And a couple of studies here that you can look up for the details to show that if you just shift the microbiota, and I've shown these diagrams here to emphasize that when you're given the antibiotics, you're not trying to destroy the microbiota, you're not trying to wipe it out. All you're trying to do for a very limited period of time is to nudge the relative abundances in a direction, just to make the point that dysbiosis alters the system. And the two systems I'm showing here, one is visceral pain perception, where you take a balloon and blow it up in the colorectum of the mouse and induce a pain response, and that's been shown to be altered by the microbiota. If you nudge the microbiota in a certain direction, you get increased pain responses. And a similar strategy using a similar cocktail was shown to alter transit, and that this alteration in transit was mediated by, TLR4 receptors. So there is this then relationship between transit and the microbiota. So slow colonic transit in humans has been associated with an increase in terms of richness and altered fermentation profiles. And on the other side, antibiotic-induced dysbiosis slows transit. So you've got this sort of yin-yang effect, between what goes on with the microbiome and what goes on with, in this particular example, intestinal motility or transit. And I think that one has to understand that this is a very dynamic situation, that if you do something to alter host physiology, either by stress or whatever, you're going to alter the composition of the microbiota, because you're going to provide a different niche for certain bacteria. On the other hand, if you directly come in and change the microbiota, you're going to alter host physiology. So it goes round in circles. And this is, in my opinion, why, for example, in conditions like an irritable bowel syndrome, that I'll talk about later, which is a chronic disease characterized by fluctuations, qualitative and quantitative, in gut physiology, it's associated with an unstable microbiota. So I think it just feeds into the chronicity. We should talk a little bit about how bacteria influence the host through their metabolic pathways. So it's been shown, for example, that the bacterial metabolism of dietary carbohydrates that produces, three main short-chain fatty acids, acetate, butyrate, and propionate, have many impacts actually on host physiology. Acetate alters cholesterol metabolism. Butyrate, in particular, has anti-inflammatory effects in the intestine. It's essential for the maintenance of the epithelium in the colon, and it's been used for a treatment, for example, in patients with ulcerative colitis whose colonic epithelium is broken down. And also, it stimulates immune function in a protective way. And it also provides, it also stimulates gluconeogenesis, which is a source of energy for the host. Bile acids, the interaction between the microbiota and bile acids is becoming an area of increasing research. So there is clearly bacterial deconjugation of bile acids, and this has effects on the microbial community. It produces different effects on different bacteria, so it exerts selection pressures, so you get a compositional change, and this alters the diversity of the microbial composition. And then, as you all know, and certainly your medical students will want to know, that the microbiota are essential in, for example, production of vitamin K from vitamin K1 dietary sources, usually leafy green vegetables, kale, and so forth. And this is important for coagulation for the host, but also it's important for bacterial function. In terms of xenobiotics, are substances that are really foreign to the body, and these are also metabolized to some degree by the microbiota. And this, I think, is very important if you're teaching medical students, because it can account for some of the issues that we see in terms of treating patients with drugs or susceptibility to certain diseases. So, for example, the degradation of digoxin in patients with heart failure is really a function of the metabolic activity of the microbiota. The degradation of L-DOPA, which is the treatment for Parkinson's disease, again, is affected by the composition of the microbiota and can account for why there are some patients that appear to be refractory to that drug. And in inflammatory bowel disease, particularly ulcerative colitis, that the cleavage of sulfasalazine, which is a commonly used first-line treatment, or at least used to be, is critically dependent on the microbiota, which converts it to 5-aminosalicylic acid, which is of therapeutic benefit. The conversion of dietary choline into trimethylamine has now been recognized as a risk factor for cardiovascular disease and is an active area of research in terms of modulating the microbiome and reducing the risk of cardiovascular disease. So this will be an important component of any instruction that you give to medical students regarding how the microbiome might affect human disease. So what about the gut-brain axis and the microbiota? So the gut-brain axis, as you all know, is a bidirectional communication system involving, how do I turn this off? Involving neural, endocrine, metabolic, and immunological signaling. The afferent part of this, gut to brain, is important in the regulation of appetite control, in the perception of pain, and in the monitoring by the brain of the inflammatory or immunological status of the gut. The brain in turn responds to this by altering intestinal physiology and through the vagal efferents, for example, attenuating inflammatory responses in the intestine, Kevin Tracy's work on the inflammatory reflex. And this is relevant, obviously, to the maintenance of health and expression of disease. And then the integration of the microbiota into this is really based on, in terms of the afferent component, demonstrations of the impact of the microbiota on the brain and behavior, and I'll show you that, as well as a demonstration for the descending pathway that alterations in brain function, particularly stress, can alter the composition of the microbiota. So what about the ability then of stress, psychological stress, to alter the microbial composition of the gut? Psychological stress, very important in almost every condition that we see as clinicians. And there are 3 models that have been published that show quite clearly that if you induce psychological stress, you can demonstrably change the composition of the microbiota. And this is done because the stress will affect intestinal physiology. And once you affect intestinal physiology, whether it's transit, motility, or even epithelial function or mucus production, you've changed the habitat, so it's inevitable that you'll get a change in composition of the microbiota. There's a couple of observational studies done in humans, using the closest thing we get to laboratory animals in humans. One medical students on the right, and then Russian astronauts in training on the left. It's been shown that when the students were taking their exams, that their microbiota changed. Uncontrolled observation. Clearly, when you're studying for exams and under the pressures that you as teachers put them under, a lot of things change, as well as stress, maybe their diet and sleeping habits and so forth. And as for the Russian astronauts working in bizarre environments, again, it's almost predictable that you'd get it. But I thought I'd just show you this to say that some attempts have been made to look at this in humans, but uncontrolled. So what's the evidence then supporting this microbiota-to-brain communication system? Sorry, this shouldn't be the effect of stress. So one is the same strategy I showed you before, the comparison of germ-free mice with colonized mice. Second is the adoptive transfer of behavioral phenotype between the different mice. And the third is perturbation of a stable microbiota and observing changes in brain function. Some people say that the demonstration that probiotics can influence brain and behavior is evidence of this. It's a little bit indirect. We recently showed a probiotic that could improve depression and brain activity associated with depression in humans. So what are the constraints in trying to do these kinds of experiments? And one is, of course, that when you're trying to characterize behavior in mice, there are many, many different apparatus that you can use, but you have to use very simple stuff because of the risk of contamination. The apparatus has to be able to withstand sterilization. The maneuver has to involve minimal handling for two reasons. One, it reduces the risk of contamination. Second, if you're dealing with germ-free mice, you're working in an isolator where the space is very, very limited. This is an example of our germ-free facility at McMaster, where, for example, when we're doing our behavioral studies on the germ-free mice on the right there, there's a drop-down, there is an isolator above where the mice live, but the apparatus for testing them is just immediately below. But the space constraints are pretty apparent. So there are a number of tests that you can do. You can do a step-down test where you put an animal on an elevated platform and measure the time it takes for it to explore its environment. And the more anxious, the longer it stays on the platform. Normally mice enjoy dark environments, and so you give them a choice in a dark chamber or a light chamber, and the amount of time they don't spend in the light chamber, another reflection of anxiety. Or you can put them on this elevated maze where their tendency to explore is tested, and they have a choice of going into a closed chamber, dark and safe, or coming out onto an open maze, and that's the amount of time they don't spend on the open maze or in the dark maze is a reflection of anxiety. And this is a study, one of the first, to show that germ-free mice, first of all, in blue here, the amount of time that that a normal mouse will spend exploring that dangerous open limb is relatively low. But the difference between germ-free mice and conventional mice is that germ-free mice are gregarious. They seem to be risk takers. They're basically uninhibited. But when you colonize them, you bring them back down to this normal behavior here. The second strategy is to take either dietary intervention or antimicrobials, and to modify the microbiota. You sequence the microbiota before, during, and after. And what we and other groups have found, and I've provided the references here, is that when you do this, when you shift the composition, you get changes in behavior, in cognitive function, in anxiety-like behavior, cognitive function, and corresponding changes in brain chemistry. Then the third strategy, which is the transfer of host phenotype via the fecal microbiota. You take a microbiota from a donor mouse, give it to a recipient mouse, and see whether you can induce the changes, or you can take a human donor and give it to a mouse, and a germ-free mouse usually, and see whether you get that mouse to colonize with the human microbiota and then assess the phenotype. And we have done this with behavior. So this strategy, when it comes to behavior, exploits two things: that commonly available mouse strains in the lab have different personality traits, different behavioral traits. In our hands, the Swiss mice are particularly aggressive, unpopular with students, whereas BALB C mice are calm, everybody's favorite. So we also know that the microbiota in these strains is a little different. So exploiting those two, what we did was take the quiet BALB C mouse, take its microbiota, colonize a Swiss mouse that typically behaves in an aggressive way, and found that the Swiss mouse with the BALB C flora behaved more like a quiet BALB C mouse. The data are shown here. This is the latency to step down. This is the Swiss mouse that doesn't spend much time because it's aggressive and explores. This is when this is an autologous transplant here, where you give it its own microbiota, or from another Swiss mouse. Again, you get this change, this kind of aggressive behavior. But when you colonize it with the microbiota from a BALB C mouse, they become much less aggressive, more conservative, and they stay on the platform for longer. And this is accompanied by changes in brain-derived neurotropic factor in the hippocampus. All this has been published. The references are there if you need them. And then in terms of underlying mechanisms, there are many, many mechanisms whereby the microbiota can influence brain function. And these are primarily immune activation, particularly innate immune activation, vagal pathways, and metabolic pathways. And the areas of current interest are in the immune system and in the metabolic pathways, particularly the microbial metabolism of tryptophan into kynurenine and other pathways as a possible factor. Trace amines are also an area of interest. But there are two things I'll say here. It's unlikely that there, in any situation, that it's one pathway. And two, when you look at the different models that are used to see the relationship between the microbiota and behavior, the models change, the mechanism changes depending on the model. So if you give a probiotic, in the studies that have been done, the benefit of the probiotic in improving or changing behavior is absolutely dependent on the integrity of the vagus nerve. Whereas if you do the other kinds of experiments that I showed, it's not. So one can't extrapolate. I think the bottom line here is that there are three pathways for this communication, and they probably differ and coexist depending on the model. Let's get to human disease. And ever since Marshall and Warren won the Nobel Prize for showing the relationship between Helicobacter and ulcer disease and also gastric cancer, there's been this explosion of interest in the microbiome as a solution for everything we don't know about human health. And I have to say that, as you probably all know, expectations are running high, and you don't have to read too many lay magazines to come across an article about the microbiota being the cause of this, that, and the other. And I'm sure that your medical students will have questions with that. And the reason for this is that a lot of this is simply based on association studies, where you take usually 16S-based profiling of a human subject, healthy, compare it to a group, often a very small group of patients with the disease, and showing that there are differences in usually small differences in relative abundance. And this has been found for a whole variety of conditions listed here. But as yet, for not one of these conditions has there been a reproducible microbial signature that we can attach to that condition. So this area of association has raised the hype, but really has not really taken the field too much further. And association doesn't imply causality, and there was a commentary in Nature that this field deserves a healthy dose of skepticism, and I couldn't agree more. So how do we get beyond association? And the answer is that to try to establish more of a sort of causal relationship. And this can be done by taking the microbiota from patients and putting it into, for example, germ-free mice to see whether you can reestablish certain components of the human phenotype in the mouse. And this is now being done extensively, and it's been done for these conditions here: obesity, metabolic syndrome, insulin resistance, models of altered behavior, I'll come to that in a minute, susceptibility to inflammatory bowel disease, although, surprisingly, nobody's been able to take the microbiota from an IBD patient and induce IBD into a germ-free mouse, unless that mouse is genetically susceptible or is given an environmental provocation to induce inflammation. Functional GI disorders, I'll come to that in a minute. Multiple sclerosis, or the excuse me, mouse equivalent. Have been done, and there is emerging data from Sarkis Mesmajian's group in Caltech that this can be done with Parkinson's disease and some psychiatric conditions such as anxiety. So I'm going to show you an experiment that was done and published recently and it addresses functional GI disorders. So functional GI disorders, as you probably know, characterized by chronic abdominal symptoms, pain, altered bowel habit, but the bowel is structurally normal. It's considered a functional disorder involving transit, motility, pain, perception, etc. It's the irritable bowel is the prototype of this condition, and it's the most common condition that we as gastroenterologists see. Its etiology is unknown. There are no diagnostic markers or biomarkers, and up to 85% of patients exhibit some degree of psychiatric comorbidity, including depression and anxiety, and as a result, clinicians often tell patients it's all in your head, go away. And it's been shown that there are alterations in the microbiota in some of these patients. Again, no diagnostic signature, but one of the characteristics of this is instability. The microbiota is very unstable in these patients for reasons that are clear if the physiology is altered. So we chose this because it was a condition that involved intestinal dysfunction and it involved brain dysfunction. And so we took patients with IBS who had no anxiety, took their microbiota and put it into germ-free mouse and, who had, let's go up here. We took IBS patients who had no anxiety, put their microbiota into germ-free mice. We were able to increase transit, because these were diarrhea-predominant patients, alter epithelial function, change barrier function, and change the secretory profile of the epithelium, but no change in behavior was seen. When we took the IBS patients who had very high levels of anxiety, we were able to produce all of these changes here, but also anxiety-like behavior. And we also saw evidence of gut function, as you'd expect, because they had GI symptoms. But in this particular group, where there was anxiety-induced, we saw evidence of innate immune activation. So at the end of the day, if we make these relationships between the microbiota and these conditions, what can we do about it? What are the treatments that are options? And the answer is they're pretty primitive. So we can try to kill off the bacteria using antibiotics, and that's really not an option, except in certain cases like Clostridium difficile colitis. We can try to starve the bacteria by reducing the amount of fermentable substrate in the diet, and this has some benefit in some patients, particularly if they're feeling bloated and distended and so forth. We can try to selectively feed beneficial bacteria, and these are called prebiotics, and it's been shown, for example, you can give certain prebiotics to enhance bifidobacteria, which is thought to be a beneficial step in the treatment of irritable bowel syndrome. We can try to supplement by giving probiotics. This is a very poorly controlled industry, as you know. Every probiotic is, the differences across the strains of probiotics are incredible. And it's a very, very difficult industry to legislate and to interpret the results. But there are a few instances where selective and well-characterized probiotics have benefit in the gut or the brain. In the future, you might try to modify the behavior of the microbiota using bacteriophage, or we can try to, quotes unquote, replace by using fecal microbial transplantation. And this is something that we hear a great deal about. Certainly the medical students that I teach want to know all about fecal microbial transplantation. Patients come to me because they want fecal microbial transplantation. It's not ready for prime time in most instances, except for Clostridium difficile, but perhaps it's something we can discuss in the, later. A couple of bugs to watch for in the coming couple of years that may have therapeutic potential. One is Akkermansia mucinophilia, because it improves barrier function, is anti-inflammatory, and Faecalibacterium prausnitzki, again anti-inflammatory. These are two commensal bacteria that probably have definite therapeutic benefit if they can be administered properly. So in conclusion then, I've emphasized the orderly colonization of the gut is critical to prevent the diseases later in life, that this is important for maturity of the immune system and physiological tissues, that in the mature host, the stability of the microbiota is critical for homeostasis in the brain and in the gut. And if you perturb it, you can change systems there. And that these perturbation or dysbiosis may contribute to the expression of intestinal, metabolic, and CNS conditions. And the establishment of causality, and I think we're a long way from that yet, will